My name is Carlos Duarte, and I'm president of ASLO. And I'm here to present today the Raymond Lindeman Award. And the Raymond Lindeman Award was created by ASLO to recognize an outstanding paper on aquatic sciences by a young scientist. And the Random Lindeman Award 2010 will be presented to Roman Stoker, who's already in the podium with me, for his paper on rapid chemostatic response enables marine bacteria to exploit microscale nutrient patches. And it was published in the Proceedings of the National Academy of Sciences. This paper uses an elegant microfluidic approach to resolve the capacity of bacteria to track patches of dissolved organic matter and quickly move towards them. And it explains the very tight coupling between bacteria and primary production in the ocean. So even though the research was done at the very microscale, it has global consequences. And I would like to say a few words about Roman before I give him the floor to present his outstanding research to you. And Roman is an assistant professor at MIT. He was born in Italy and received his BSc and his PhD from the University of Padova in Italy, and his PhD was on lake hydrodynamics. Then he moved to a number of postdoc positions, including one in the University of Western Australia in Perth, and since 2002, I believe, no, 2005, Roman has been at MIT, where he is now assistant professor. And I don't want to steal more of his time, and before he goes along with his talk, I'd like to present him with a much-deserved award. So congratulations, Roman.
Thank you very much, Carlos. It's an honor and a pleasure to be here. It's an honor to be receiving this award today, in particular because I've started in this line of research only 5 or 6 years ago, and it's really fantastic that these ideas and these efforts are being recognized by the community. In 1959, Richard Feynman gave a talk at Caltech entitled "There is Plenty of Room at the Bottom," where the bottom included scientific questions at the microscale. He was addressing an audience of physicists discussing open problems in biology. It is very easy to answer many of these fundamental biological questions, he said. You just look at the thing. He didn't mean to belittle biology by saying that. He was spurring physicists to develop techniques to look at biological processes at very small scales. Today I will give you a couple of examples of biological questions at small scales at the bottom of the ocean, where with bottom I don't mean great depths here or sediment-water interfaces, but in the same sense that Feynman intended it, the microscale, the microbes and the microbial processes. And I would like to argue in the process that we are now in a position for many of these processes to really just look at the thing. But before we get there, what happens at the bottom of the ocean? All sorts of vital functions, to the point that it is now believed that much of the biogeochemistry of oceans and lakes is really ruled by microbes, and heterotrophic bacteria play a fundamental role in that. One essential function that they perform is the recycling of dissolved organic carbon. This carbon is spit out by phytoplankton. It's dissolved and therefore would be lost from the system unless organisms like bacteria could take it up. The bacteria then become food themselves until the carbon is returned to the food web. This yellow loop of arrows is called the microbial loop. This, however, is just a flow chart. What do these processes actually look like? Turns out we don't fully know, but a picture that is beginning to emerge is one in which these interactions are not uniform. They don't occur like in a homogeneous soup, but there are plenty of processes that structure the environment at very small scales. And this image would like to show some examples of that. Excretions by larger organisms like zooplankton, phytoplankton that exude dissolved organic carbon, lysis of phytoplankton cells, or marine snow particles that sink and generate these intense plumes of nutrients. In this heterogeneous milliliter of water, many bacteria, although the number is difficult to quantify at present, have evolved the ability to swim using these helical flagella. They can sense their chemical environment, and presumably they might be able to take advantage of these micropatches. Is this a plausible hypothesis? Well, Feynman would tell you, just look at the thing. So this is what we tried to do. We use microfluidic devices, which are small-scale chips on which you can accurately fabricate chambers and channels. You can create flows, and you can control precisely the chemical landscape. You can then stick the whole thing under a microscope and simply visualize what the organisms do in response, essentially extending to microbes what ecologists have done for larger organisms for a long time. So in the paper, we've used this approach to study a couple of important processes in the ocean. The first one has to do with that algal lysis process that I told you about a couple of minutes ago. This microchannel is designed to mimic a one-dimensional analog of that process, and it works in the following way. We have a first inlet where we inject algal exudates. They are then squeezed out through this little microinjector with a process that is shown here in this movie. At the same time, we have a second inlet for bacteria. Bacteria are transported by the flow so that they find themselves adjacent to this layer of algal exudates. And we then do a very simple experiment. We just stop the flow, which mimics the release by the dying alga. What happens in that case is that transverse to the channel, the exudates will diffuse out, as you would expect. But most importantly, we're now in a position to visualize what the organisms really are doing at the scale at which they're doing it. Watch what happens in the second image here. Each white wiggle here is a trajectory of a single bacterium. We have thousands of them here, and many of them have located this algal exudate band and have accumulated within it. To give you a sense of just how rapid this response is, we compared it with the most rapid response of a model organism, E. coli, which is the reference organism for motility in chemotaxis. And you can see here that the marine bacterium that we used vastly outperforms E. coli, even under its best conditions, both in terms of how strongly it accumulates in that band, which is measured by this hotspot index, as well as how rapidly it does so. This, we calculate in the paper, gives the marine bacteria a 10-fold larger nutrient gain, and we speculate that it might be an adaptation to the typical nutrient environment that organisms would find in aquatic environments. From a technological point of view, we found this platform, both in terms of gradient generation and visualization, to be very successful, and we applied it to a number of other questions since then, which I won't have time to go into now. The second process that we studied in the paper is the coupling between the movement behavior of the organisms and these plumes of organic matter that I showed in the diagram earlier. These particles, often referred to as marine snow particles, play a fundamental role in the carbon cycle because as they settle, they transport carbon to deeper depths. And marine bacteria interact in very interesting manners with these particles. Dense populations of bacteria reside on the particles and solubilize them, and the dissolved carbon that results from that process leaks out the back. Can bacteria in the surrounding water column take advantage of these plumes? Well, an elegant mathematical modeling study by Thomas Kirby and George Jackson, published in L&O a few years ago, suggested that under certain conditions they do, but at the time there was no way to just look at the process. That's when we came up with the idea of trying to simulate that with the microchannel, where we can recreate particles of the appropriate size and appropriate sinking speed, in addition to the plumes that they produce. Look at what happens to the population of bacteria in this case. Each dot represents a single bacterium. As they first encounter the plume, they're nearly randomly distributed. When you move only a few diameters downstream, by 3 or 4 millimeters downstream, most of these bacteria have located the gradients and they swim into the center of the plume, providing the first observation that these coupling can actually occur. And in the paper we computed this gives them a 4-fold advantage in terms of nutrient uptake compared to species that do not have motility. So there's a couple of things I'd like to mention before we close. There's two elements that we need to include, or two general elements that we're trying to include in order to make this story more general. The first one is turbulence, which is of course ubiquitous in oceans and lakes. We're tackling this with direct numerical simulations in which we can understand what the role is of turbulence on these dissolved organic matter patches, as well as on the ability of organisms to take them up. The second point is the realization that the world in which these organisms live is truly a world of gradients. But these gradients are not just gradients in nutrients, they're also gradients in flow, in particular shear and shear forces. And we're finding that these shear forces have an overwhelming effect on the swimming and accumulation of the organisms, and ultimately significantly affect the ecological interactions. So in closing, I'd like to come back where we started from. The wide diversity of microbes out there, the vital functions that they play in the ecosystem, as well as these really interesting interactions with flow gradients as well as nutrient gradients, truly suggest that there is plenty of room at the bottom in research in oceanography and limnology. One way to go about this is, of course, the molecular avenue, which is rightfully receiving a lot of attention. But I would like to argue that there is plenty of room in terms of discovering functions and processes also by just looking at the thing. In closing, I'd like to thank the collaborators on that paper, in particular former postdoc Justin Seymour, now in Australia, Martin Polz, a professor at MIT, Dana Hunt and Azadeh Samadani, funding from the National Science Foundation. And I'd like to shamelessly take 5 seconds of your time and leave up this last slide for advertising a meeting that we're organizing, much on similar themes, at the Aspen Center for Physics in January of 2011. Thanks again for the award, and thank you very much for your attention.
Okay, I'd like to just do one more round of applause for all the speakers, Tara Pleewell, John Haynes, John Davis, Roman Stoker. Terrifically interesting morning to me. I hope you agree, and it bodes well for the rest of the meeting.